Hello, everybody. Welcome to today's video lecture, and we're covering muscle tissue. Let's go ahead and begin. So functions of muscles, movement of bones or fluids. So any type of movement, you can move your entire body, right? You can move your fingers, your arms, you can walk, you can talk, moving your tongue, or some fluids in your body, like your blood can move through your blood vessels, or chyme moves through your digestive system. It's not fluid, but it's your it's your food partially digested and mixed with digestive juices, right? Another function is maintaining posture and body position, stabilizing joints, and generation of heat, especially skeletal muscles. Skeletal muscles, or actually all muscles, and we have three types of muscles. We have skeletal muscles, we have smooth muscles, and cardiac muscles. They have their special characteristics, such as excitability, so its ability to respond or being irritated, so irritability, so ability to receive and respond to stimuli, contractility, ability to shorten when stimulated, extensibility, ability to be stretched, and elasticity, or ability to recoil to resting legs, right? So when we talk about muscle functions and special muscle characteristics. And I already mentioned we have three muscle tissue types: skeletal, smooth, and cardiac. Now here we can briefly compare these three types of muscles by their location and by the cell shape and some other characteristic mentioned in this table. Right, so skeletal muscles attached to bones or some facial muscles attached to your skin. Cardiac muscles found only in walls of the heart, and smooth muscles, they found in the walls of hollow organs. So here, walls of hollow visceral organs, other than the heart, because we have cardiac muscles in your heart. What does it mean, your hollow organ? So for example, your GI tract, right, including your stomach, including your small, large intestine, or your urinary bladder, or your blood vessels, right? So those are hollow organs, and in the walls of these organs, you will find smooth muscles. Also, you will find them, it's intrinsic eye muscles, also made of smooth muscle tissue. Right, so if you're looking at a shape of muscle cells, then in skeletal muscles, they are cylindrical, very long, multinucleated, with obvious striations. So if you look at this diagram over here, so those long fibers are individual cells, and those dark parts are nuclei. So we have several nuclei within one long cell, and those lines that you see over here, they're called striations. So those are striations. Now, because skeletal muscles are thin and very long, sometimes we call them muscle fibers. So muscle fiber is the same as skeletal muscle cell. Cardiac muscles, they are branching chains of cells, uni or sometimes binucleated, and they also have visible striation. So for example, over here, if you see, cells, and they kind of have these branching parts, right? For example, right there, maybe that would be a cell and has this some, branches over over here, right? So that's the nucleus of a cell. Now, these lines over here in darker color, this is special structure found in cardiac muscles called intercalated discs. And those discs are connection between two adjacent muscle cells. And you can see striation as well, right? So over here. Now, smooth muscles, they are spindle shape. So you can see over here, spindle shape. They're uninucleated and no striation. So again, this dark part you see is nucleus, and then, you know, cells surrounding this nucleus. Right. Oops. So those are smooth muscle. And another characteristic you want to remember that your skeletal muscles are voluntary. That means you consciously can control their contraction and movement. So they're voluntary. And cardiac and smooth muscles are involuntary. That means you cannot control those muscles, and they will be controlled by your autonomic nervous system. Now, how muscles are attached to your bones or to your skin? We have direct attachment and indirect. So direct attachment is not very common. It's when we have epimysium of the muscle, and epimysium is the connective tissue covering entire muscle. And that connective tissue is fused to the periosteum of a bone or perichondrium of cartilage. And periosteum, perichondrium are connective tissue covering bone or cartilage. Right, but that's not very common type of attachment. More often, muscles are attached to your bone indirectly. When we have connective tissue wrappings extend beyond the muscle as a rope-like tendon or sheet-like aponeurosis. Right, so you can see over here, direct attachment are less common. Epimysium is fused to periosteum or perichondrium. And over here, muscle attachment may be direct or indirect, right? And here, our indirect attachment show us we can have a tendon, right? So tendon shown here in the white color. So in pink color represent muscle. And then we have this connective tissues that extend beyond the muscle, right, as a cord-like structure, then it's a tendon, or it can be a sheet-like structure, like shown over here, for example, right? So all this structure here, we call it aponeurosis. And then tendon or aponeurosis attached to periosteum or perichondrium. Now let's look at organization of skeletal muscles. So over here we have what it's called muscle fiber, and muscle fiber is the same as muscle cell, right? So we have individual cells that are very long, cylindrical, multinucleated. So you can see nuclei over here, right? Now, individual muscle cells are covered by connective tissue. So you can see okay, this might just represent cell membrane. I'm sorry, yeah, sarcomere. Sarcomere is just a cell membrane. So we have just a cell membrane, right? Inside cell membrane, we have nuclei and all organelles, different organelles. So over here, you can see mitochondria. I already showed you nucleus, and those are protein fibers, microfibrils, they are called. So we have lots of protein fibers, bundles of protein fibers inside a muscle cell, right? So over here we have those, muscle fibers, and then those muscle fibers are covered by connective tissue called endomysium. Endo means inside, so we have every cell covered by endomysium. Right, so you would say that's a muscle fiber, and endomysium would be, this kind of pinkish structure over here, right? So that cover individual muscle cell. So endomysium contains extracellular fluid and nutrients. All right, then if we have those dark spots over here represent individual muscle cells, they covered by endomysium, and then they organized into bundles. So here is a whole bundle, right? And this bundle of cells now covered by perimysium. And this bundle is called fascicle. So we have fascicles, right? And those fascicles, so for example, this is a fascicle, this is fascicle, this is fascicle, right? And this is fascicle. And of course, this is the fascicles that it magnified for us to see. What is inside this fascicle? Individual muscle cells, right? Now, fascicles are covered by perimysium. And then we have, lots of these fascicles covered by perimysium that form entire muscle, and entire muscle is covered by epimysium. So it wraps entire muscle, separate it from other muscles, allow contraction while maintaining stability. Perimysium surrounds fascicle, common in limbs, allows movement of specific muscles, and endomysium contains extracellular fluid and nutrients, and then what covered by endomysium are individual muscle fibers or cells. And aponeurosis, we just mentioned, or another name is fascia. It's a connective tissue between skin and the skin and the bones. That's just wrong. What is aponeurosis? We already know, right? It's a sheet-like structure that attaches muscles to the periosteum or perichondrium, right? And we do have fascia. So we have like superficial fascia that is under your skin, but it's between your skin and your muscles, right? So I don't think this is a correct sentence here. Now let's look inside one individual muscle cell, right? So here we have a muscle cell, right? And as any cell, it must have cell membrane, right? Because there is no cell without cell membrane. Cell membrane separates cytoplasm and nucleus from extracellular fluid. It separates what is inside of a cell from outside environment. So we must have cell membrane. However, cell membrane in skeletal muscles called sarcomere, right? It has a different name, and it has different name for a reason, because the structure is different from a regular plasma membrane surrounding, for example, your skin cells. Now, inside we have organelles, right, and cytosol, fluids, organelles, and fluids and organelles are called cytoplasm. Now, fluid portion of cytoplasm called cytosol. However, in the muscle cells, we call it sarcoplasm. Now, this sarco means muscle. So we have sarcolemma and sarcoplasm. And then we have endoplasmic reticulum. It's a specialized endoplasmic reticulum that contains lots of calcium. And it's similar to smooth ER, but still has differences. That's why this one is called sarcoplasmic reticulum instead of endoplasmic reticulum, right? So here we have it. We have sarcolemma, we have sarcoplasm, we have nuclei, and we have several nuclei in skeletal muscles. Now, part of sarcoplasm, mitochondria, other organelles, sarcoplasmic reticulum, and then we have those fibers. Those are proteins. They called myofibrils, right? And myofibrils are made of several structural proteins. The most abundant are actin or thick. I'm sorry, should be thin filaments, not think. Okay, right. So we have a thin filament and we have thick filament. Okay, I fixed it over here, but on your PowerPoint, it's still gonna be thin. Just go ahead and remove this K. So we have thin filament or actin and thick filament or myosin, right? So myosin is thick, actin is thin. So, and if you look at this myofibril, right? So this is myofibril, and we have many myofibrils inside one muscle cell. And then you see those lines over here, some shown in the green color over here, some in the purple. So the ones that shown in the green color, those are our actin. Let me see. Yeah, it's labeled right here, thin or actin. So those are these green lines, and those purple lines are myosin or thick filament or myosin. So when we, so here's a better, little bit better diagrams that show us actin, myosin, and sarcolemma. So that's a sarcolemma, that is cell membrane, right? Now look how the sarcolemma, it has this indentation inside, right? And this is called T-tubule. So that's a T-tubule. And next to T-tubule on the both sides, we have this blue, shown here, blue structure, right? And this is a sarcoplasmic reticulum. So all this blue structure is sarcoplasmic reticulum that stores calcium. But this part adjacent to T-tubule and this are called terminal cisterns. The name is right here, terminal cisterna. And these three structures, two cisterna and one T-tubule called a triad. So here we have triad. And it will be important when you learn about physiology of muscle contraction, right? So we have sarcolemma, we have indentation within sarcolemma called T-tubules. Next to T-tubules, we have terminal cisterns, and two cisterns and one T-tubule form triads. And you can see the sarcoplasmic reticulum kind of surrounding our filaments. This one is our myosin or thick filament right here. And this shown in red is actin or thin filament. Now, if you look over here, you see this structure look like Z line, right? It's actually used to be called Z line. Now they call it Z disc. So those are also structural proteins within those myofibrils. So this area from one Z disc to another Z disc is called sarcomere. And we have repeating units, repeating sarcomeres within those myofibrils. So you can see here's one sarcomere over here, is another sarcomere, right? Now the sarcomere is a functional and structural unit of skeletal muscles. And on this diagram, you can see sarcomere in a detail. So here's our sarcomere. This is our, you see it's called Z line over here or Z disc. And what is attached to Z line is actin. Then inside the sarcomere, we have M line, like kind of in the middle, M in the middle. And what attached to M line are myosin, right? Let me just remove all my drawings here. Right, so again, from one Z disc to another Z disc, or Z line, we have one sarcomere that is functional and structural unit of muscle fiber. Proteins attached to Z line are actin. Proteins that attach to M line are myosin, right? And there is some bands and zones that you need to know of the sarcomere. Now look, where you have only thin filament, only this green filament. It's called I band. And I remember it like I is very thin letter, right? So that's I, right? I band is only actin. Now, where we have, so this area that includes myosin. So it's completely as long as this myosin molecule in the middle, right? So if you have myosin here, right, this area is called A band. And you see A band includes a little bit of the actin as well. So here we have actin, this green part. So A band includes what? Both myosin mostly and actin, right? We have myosin and actin in A band, and we have only actin in I band. And where we only have myosin, we call H zone, right? H zone. So let's kind of, review it just one more time. So what is a sarcomere? It's the area between two Z lines or two Z discs. Now, the area where we have only actin is called I band. The area we have only myosin is called H zone. So you can see H zone, we have only myosin. And the area that equals in length to myosin and include both myosin and actin is A band, right? So I band, because we only have actin, it's appears lighter. A band, because it has a lot of myosin, it appears darker. So you see how we have lighter line, darker area, lighter area, darker area. And this is actually what make striation of the muscles. So you see how over here we have lighter, darker, lighter, darker. And if we move back over here, you can see in a skeletal muscle over here, right? How we have darker lines, darker, lighter, darker, lighter. And the same in cardiac muscles, darker and lighter lines. So now we know what those lines represent actually, and what those lines represent I band and A band. I band, A band, I band, A band, right? Okay. Now, over here, you can look at structure of myosin. So it has a tail, it has flexible hinge region and two heads. And in the area of the head, you have ATP binding site, and this is where ATP molecule binds, right? And we need ATP for energy. If we want to contract muscles, we need to use ATP, right? So ATP has its own binding site. And also on the head of myosin, we have actin binding site, right? So myosin wanna bind to actin and actin wanna bind to myosin. And when we have this binding and we use ATP, we can pull actually this actin towards M line and shorten the sarcomere. And this is how your muscle shortens when they contract, right? But this will be explained more in physiology. So anyway, myosin has a, so we have myosin molecules that has a tail, hinge region, and two heads with actin binding site. Over here, you can look at the structure of actin. Now, here's we have a actin subunits, right? And each subunit has myosin binding site. So myosin wanna binds to actin, it has actin binding site, and actin has myosin binding site. So these two sides can bind together, produce shortening of the sarcomere. But if you look at this binding site, it's actually blocked. It's blocked by a molecule that is called tropomyosin. So this orange molecule over here is tropomyosin, and then this molecule over here is troponin. So here's the troponin. So we have actin, we have tropomyosin and troponin, and this prevent actin from binding to myosin, because you don't always need actin and myosin to bind. You don't want your muscles contract constantly, right? That's why you have this blockage. And when calcium is present, calcium binds to troponin, and then it moves tropomyosin and exposes this binding site, and it can bind to myosin, and we can have contraction of muscle and shortening of sarcomere and contraction of muscle, right? So sarcomere is functional unit of a muscle fiber. It's a region of one Z line to the next Z line. Thin filament is actin, troponin, tropomyosin, not action. Actin. I'm sorry for this, actin plus troponin plus tropomyosin, and thick filament is myosin, right? Okay, so good. Okay, now, and again, we will talk about muscle contraction in more detail in physiology. So let's see how muscle develop and regenerate. So actually, during embryonic development, we have cells, myoblast cells that can divide and make more and differentiate into your muscle cells. But when you are born, you actually have your muscle cells, muscle fibers that cannot divide anymore, right? So you're born with all the muscle cells that you ever will have. However, your muscle can still get bigger. So how can your muscles get bigger if muscle cells cannot divide? So for something to get bigger, if it's made of cells, right, we usually want cells to divide, make more cells, and the organ or organism is getting bigger. Now, muscles are getting bigger, but muscle cells cannot divide. So how is that possible? Because you actually have what it's called satellite cells. They're similar to myoblasts. So here's the satellite cell shown right here. And those satellite cells, they can incorporate themselves into muscle cell. And satellite cells, they have their nucleus, right? They have all their mitochondria and Golgi apparatus and endoplasmic reticulum. They have all those, and ribosomes. They have all these organelles. So when we have incorporation of satellite cells inside a muscle cell, this cell deposit its, nucleus and a cytoplasm right inside your muscle cell, and this facilitate protein synthesis. So we have more DNA, we have more cytosol, and in the cytosol you will have amino acid and other important molecules such as enzymes, and you have your ribosome. So this muscle cells start synthesizing more proteins, and when we have more protein inside the cell is getting larger. So that's called hypertrophy. So it's the same cell, right? It's one cell, and this is still one cell, but this cell is larger because it has more organelles, it has more proteins inside, right? Right, so this is how your cells are getting larger, right? And this is how they can regenerate, partially regenerate. So actually this diagram show you the difference between muscles that were trained and the ones that were not trained. So those are untrained muscles. Then you have first training root, and you have fusion of satellite cells with muscle fiber. They go through hypertrophy. If you stop training your muscles, detraining, you will get to atrophy. All right, but however, you still, now you have more higher potential, right? You have more nuclei, you have more mitochondria and ribosomes, so it will be easier to retrain this muscle to get it back to a bigger, a larger shape, right? So anyway, this is how muscle develop and regenerate. Sometimes when we damage muscles, satellite cells, so when you damage muscles, satellite cells partially can, regenerate your cells, right? So they can repair. But if damage is extensive, right, then muscle cells will heal by fibrosis. And that means that muscle fibers will be replaced by scar tissue. All right, so, and if you have a deep cut in your muscles, you will have scar developed, right? Because muscle cells cannot divide, they cannot really fully regenerate. Very often they heal by fibrosis. Parasite are stem cells that regenerate smooth muscle. So out of 3 muscle types, we have skeletal, we have smooth, and we have cardiac muscles. The smooth muscles are better at regeneration because they do have parasites that are capable of division, and they can replace damaged cells. Now, muscular dystrophy, what is that? It's a group of inherited muscle-destroying diseases. During muscular dystrophy, muscles enlarge due to fat and connective tissue deposit, and muscle fiber will atrophy. So let's see what's happening in your muscle cells. So this is your cell membrane or sarcomere of a muscle cell. So this is cytoplasm. Inside, right? And this is extracellular fluid over here, and extracellular matrix. We have proteins in the extracellular matrix. Now, embedded in the sarcomere are different membrane proteins, and some of these proteins they anchor the cell to the extracellular matrix and to the cytoskeleton. So you can see this protein over here, one part of it attached to extracellular proteins, extracellular matrix, and another part attached to cytoskeleton. And it gives this cell membrane stability. Because you can imagine if muscle constantly contract, right, it put a lot of force on this cell membrane, and it can break, right? It can be damaged because of, frequent contraction. But you can see to have this nice connection, we need special protein called dystrophin. So when we have muscular dystrophy, this protein is completely absent. So the person does not synthesize dystrophin, or it might be not fully well, it might be present, but because of mutation, it might not be precise shape of this protein that will not allow this nice, strong connection. Right, so either dystrophin is damaged or dystrophin is absent from a cell, then the person would have muscular dystrophy. So we have different types of dystrophy, and you can see main area of muscle weakness in different types of dystrophy. The most severe is Duchenne dystrophy over here. So Duchenne muscular dystrophy, or DMD, is most common and severe type. It's inherited, sex-linked, carried by females. So it means females carry mutation for dystrophin synthesis on the X chromosome. And when mother passes her X chromosome to her children, then children can have muscular dystrophy. And more often, as all sex-linked diseases, more often males are affected more often than females, right? Because of complete lack of dystrophin. That's why it's very severe. So victims become clumsy and fall frequently and usually, unfortunately, die of respiratory failure in their 20s. And they have respiratory failure because your diaphragm is a skeletal muscle, and muscular dystrophy affects skeletal muscles, and you need a diaphragm to breathe, right? So there is no cure for this disease, but we have some viral gene therapy or infusion of stem cells, which correct dystrophin genes, and it shows some promise, right? So over here, you can see direct and indirect effect of this condition. So direct effect will include skeletal muscle degeneration, inflammation, cardiac or respiratory dysfunction, ultimately leading to death at about age of 20. So very severe condition, and luckily now pregnant females can be tested for many inherited disorders, including DMD. And indirect effect will include mitochondrial dysfunction, impairment in autophagy, and alternation in angiogenesis. Alterations. So alteration in angiogenesis means that angiogenesis is development of blood vessels and growth of blood vessels. So it might have some, problems with development of blood vessels, and impairment in autophagy leads to accumulation of some free radicals in the body that also obviously decrease overall health of the patient, right? And I think it might be, oh no, we have two more slides to go. So we will talk about cardiac muscles. Now, cardiac muscles, we already mentioned, found only in heart. They also striated and they have sarcomeres. We have that we talk about it, intercalated discs that allow cardiac muscle cells to contract in a wave-like pattern. So intercalated discs, they connect to adjacent cells together. And desmosomes anchors the ends of cardiac muscle fibers together. So here shown in white is intercalating disc, and they have gap junctions. So those are channels that ions can go through, and they have a desmosomes, right, shown over here, that holds cells together. Cardiac muscle cells are also rhythmic cells. That means that we have what we call pacemaker cells that control contraction of the whole heart muscles. Also, rhythmicity means you don't really need innervation from central nervous system, so those cells can contract on its own. So they can generate their own electrical signals and contract in a unison, right? So in a wave-like pattern. And the last slide is about smooth muscles found in walls of hollow organs. They spindle-shaped fibers, we already mentioned it before, no striations, but they do contain thick and thin filaments. And right, so you can see on histology of smooth muscle over here, and a diagram on the top. An example, you're giving the stomach, right? That's your hollow organ, and some intrinsic muscles of your eye. Now, those smooth muscles and cardiac muscles are involuntary. That means you do not control those muscles, and they controlled for you by your autonomic nervous system. Okay, so that's it for anatomy of muscle tissue. Right, thank you for watching, and I hope it was helpful.